Good evening. Welcome to our first presentation in the Soil Health Series. Tonight, Dr. DeAnn Presley is going to talk to us about soil structure. DeAnn is a professor of environmental soil science and management in the Department of Agronomy at Kansas State University. She conducts research and extension on soils spanning a range of environments, including both agricultural and suburban. Her current focus is on the topic of cover crops and the benefits that they provide, namely on how cover crops affect soil health and crop production. Tonight, she's going to talk to us about soil structure, the importance of soil structure, and how that changes with management. She'll cover soil physical properties, including aggregate stability, bulk density, and infiltration, and its role in water and nutrient cycling. DeAnn, it's all yours.
All right, thank you. Well, thanks, Gretchen. It's great to be here. As she said, I'm going to talk about building better soils and soil health, and with a real focus on soil structure for this presentation. As she said, I'm going to focus on soil physical properties tonight, and I always like to think about building better soils. So, soil health has been defined as the capacity of a soil to function. So there are measurable chemical, physical, and biological properties that we can take in soils. And my focus is very much on the physical properties. But I think about soil health like a three-legged stool. There's the physical, the chemical, and biological. And a soil doesn't function well if it doesn't have good properties in all of these properties ranges. And so I also think about soil organic matter as kind of like being the seat, because it is a chemical property that we measure in a soil testing lab, but it's produced by the activity of biological organisms, and it has so much impact on the physical properties. So I always say yes to that question. It's kind of a property that's overarching across everything. And then how long does it take for a soil to change? And I often think that that depends on the property that you're measuring. So soil organic matter is something that is kind of a slow property to change in terms of a measurable change, but there are other properties that change even faster, and they're very important for how the soil functions too. So here's just an example of some of the soil functions. You can cycle nutrients, so nitrogen, phosphorus, carbon, and sulfur all cycle through various types of phases to where they're maybe not plant available for an NP and S, but then there are plant available forms, and it's microbes that are cycling those. You have water relations. So when rain comes in central Kansas and western Kansas in particular, we need to be able to try to store as much of that as possible. And if you get too much rain and it overcomes the infiltration capacity of a soil, then we end up with runoff. And runoff, no matter where you are, is a bad thing because it'll usually carry away sediment, it'll carry away pesticides. And so we wanting to keep the soil as open to infiltration as humanly possible. Diversity and habitat is something that soil provides to the organisms there. So it's a habitat, and the better the habitat is, the more hospitable it is, you'll get greater diversity of the organisms. And then soils also filter and buffer. So if you think about if there were a septic system in this soil, the effluent that goes into the soil, the water is degraded or detoxified, the substances in there degrade, and that's again being driven by microorganisms before anything could reach the water table. But that's not septic systems aren't the only thing. Of course, that could be true of any substance. So nitrates and any pesticides that we use in agriculture. Soils do a very important job of filtering and buffering any of the water as it goes through soil. So where do soils come from? I teach a class about soil genesis and classification, so where they come from. And one of the things that we talk about are the 12 soil orders. So mollisols are one of those 12. And I always explain to my students that mollisols are the best soils. And so Kansas is a very fortunate in that regard that we have two kinds. So we have a lot of Udalls and Ustalls. Udalls are on the more humid side of the state, and the Ustalls are in the western 3/4 of the state. And these are wonderful soils, and it's no coincidence that the amazing grain production area of the United States has mollisols present. So their mollus means soft in Latin, so they're soft soils, but they're soft because of organic matter. And so organic matter, because of the prairies have grown for many, many years, the roots have decomposed and died, has really created this amazing medium for plant growth, if you will. There are the 5 soil forming factors though. So climate is a factor. So as you go towards the desert, we don't have as much soil organic matter, just less production of plants leads to less organic matter. You can also have rapid decomposition of organic matter. So as you get hotter and wetter environments, organic matter decomposes faster. But there's also a factor of organisms. So where we have prairie plants, we have mollisols. And so much of the rest of the United States has trees. And so they just don't get as much addition of organic matter into the soil when you have trees as the organisms. Relief and parent material and time also have a lot to do with the soil. So, the flatter your landscape is, there'll be less tendency for erosion from water anyway. Parent materials, that's a whole long discussion we could have about whether it comes from glaciers or whether it comes from wind derived. And then time. So it does take soils time to form. So it took them time to form. It also takes time to rebuild the soil if it has been a bit degraded. So what is the difference between these two photos? These are under the same lighting. So there's nothing funny happening with the lighting. I haven't fiddled with it in any way. These are under the same lights in my laboratory bench. And I say that the hint here is about 150 years. So the soil on the right is a prairie, and the one on the left is cropland right over the field, or right over the fence. And so a lot of places that you go throughout the Great Plains, you'll find that cropland is about 40 to 60% eroded relative to the adjacent pasture. So erosion is just a straight up loss of soil, and it's not just that dark color, but anything else that could have been there. So any other nutrients that could have been carried away or blown away, that's also just lost from the soil with erosion. So I clicked by a little faster, but humans really are the 6th soil forming factor. We have a lot of effect on soils in a short amount of time. So that's sort of the bad news, but the rest of the time, let's talk about the good news. And that is that researchers have shown, myself and a lot of other people have shown, researchers have shown that we can improve soil health. It's possible to take it the other direction, to go back in the direction of improving it. And big picture, I just saw a new paper came out this week. Big picture, better soils equal better productivity for crops. It's not a surprise, but that is something that I think is on every producer's mind of like, how will this affect me? If I put effort in to improving my soil, how will that pay me back? There are some other short-term things too, though. Water relations. If it takes water to produce a cover crop, how does that affect my soil? Weeds. I know that's probably the number one question right now is about weeds and cover crops. So how can I control problem weeds with cover crops as part of my integrated pest management system? And then the question about how fast will my soil change? That is something that's difficult to answer, and it really depends on where you are, but it does take a little time. I think about it like putting money in your savings account. There's such a thing as interest, right? So it can pay interest, or, even better in a mutual fund, it pays interest, but you don't put it in there one day and then expect to have a lot more money the next day. It takes quite a while, but then it can really the snowball can get rolling. So the principles of building up soil properties have to do with, there's a few particular principles, and one of them is reducing soil disturbance. So that would be either eliminating tillage entirely or reducing tillage to the bare minimum in a system. So the more we disturb a soil and fluff a soil, that introduces oxygen, it changes the porosity, and then microorganisms, but then basically I think about it as cannibalizing. They'll cannibalize soil organic matter, and that's called respiration. They respire it even faster. So that change in dark color, from dark color to light color that I showed in that cropland versus pasture, it's a combo of those two things. One is the erosion taking you completely away, and two is by increasing respiration so that organisms are breaking down what's there even faster, and so that double-edged sword. Also covering the soil surface. So whether that's more crop residue, stopping baling crop residue, leaving plenty after grazing, or more cover crop, like growing cover crops so you have more cover. More cover is better for soils. And then increasing the diversity of the cropping system. It has been shown to increase soil organic matter and improve soil properties in general. So whether that's adding, going from a monoculture to 2 crops, that's definitely better. And adding a 3rd crop can be good too in a cropping system. Or if you don't want to do that, you could add a cover crop as that extra crop in your cropping system. And then adding to the carbon to the system in general. So more plants, so more days of the year, maybe if you have cover crops, or you could add manure or compost, as those are also really good sources. So I always think about how soil and microbes and plants are really connected, and I stole the recycling symbol to indicate this. But soil and plants, we know that plants use soil, where they penetrate their roots, and they anchor there so it'll blow over. And soil is a huge storage capacity for both water and for nutrients. And then plants and microbes are very related. So if you have a mycorrhizal association between mycorrhizae and plants, then the plant will provide the mycorrhizae with carbon, and the mycorrhizae might turn around and provide phosphorus to the plant. And there's a lot of people studying these relationships, and I think there's a lot of information being generated right now as to how many services are microbes providing for plants. So it's a very fascinating area of research. And then microbes in soil, of course. Microbes live in soil, it's their habitat, but then they also are little engineers of their habitat, and they will improve soil structure to have even better habitat, which is beneficial then for water getting into a soil, gases diffusing in and out of a soil. So it's win-win-win. So I'm going to talk about the importance, and I'm going to talk about some of the things we can measure. And just in general, the theme is about reversing the effects of degradation of soil. So mean weight diameter, when I talk about aggregate stability, sometimes I want to make sure I give some examples of this. Mean weight diameter is like the grade point average of your soil. So like if I were to take a soil sample, in fact, I have some here on my desk. If I were to take a soil sample and I break this up, and, I grind this all up. Usually we grind things up to 2 millimeters or whatever. I grind it all up. And then I take this and I can take a known sample, usually 50 grams, and then I'll put it on a nest of sieves where you get the big sieve on top, and then it goes down in size to the little one on the bottom. And then you dunk those up and down in water for 10 minutes. Then the mean weight diameter is basically saying how many A's and B's and C's and D's did you get? So that just like your grade point average in school was to say, a way to sort of summarize how you did in all your classes. That's like mean weight diameter. So the bigger the number, the better. Just like you want to be closer to a 4-point in school, or you hope your kids have that on their grade cards or whatever. But you want to have a higher. So mean weight diameter, the bigger the millimeter, the better. That was probably an over explanation, but here's an example from Wichita from spring 2015 when we went and sampled. So to set this up a little bit, this graph, the y-axis over here should be in millimeters. So bigger is better on this one. And in this system where it says tillage, this is tilled every spring and every fall pretty vigorously. And then for these others, there is still actually tillage in the system. So the crop grown here is pumpkins. And after the pumpkins are harvested in, September, October, then they'll go vigorously till this up. And we were planting cover crops right after that. So the choices of cover crops in the fall, we planted rye or rye and pea or rye and vetch or rye and canola or a rye vetch canola 3-way mix. And then sometime we had one set of treatments where we're like, well, what if we couldn't get in and get our cover crop planted in the fall, but instead we planted the cover crop in the spring. So oat or oat pea would be our choices there. So the point is that even in a system that has tillage in it still one time a year, the difference the cover crops made for improving a soil. So that is another thing, is that some people will say, well, I still want to do tillage, but what about cover crops in a tillage system? Cover crops are good for soil, for improving a soil, no matter whether you till or not. But again, no-till is something that'll, if your whole purpose was to improve soil structure, the best thing you could do is to do no-till and to grow cover crops. Those two things together have a really positive effect on soil. But my point is that these cover crops in even a short term, even in a very disturbed system, had a profound effect on improving soil structure. And here's the statistics on that. So there was no difference between them, but they were all better than the tillage. So this is what it looks like if you do a little slaking thing. You can take this is one where I had 3 different soils, all just right in the same area. So conventional, a no-till field, and a native prairie remnant. This was in the central Kansas by Niles, Kansas, where I got these. So you drop them in, and this is called the exploding aggregate test. So you drop them all the way in soil and see what happens. And if you don't have any structure, this will just blow apart. Because if you don't have structure, then there's just a bunch of little air pockets in there that are entrapped. And so it's like having a weak submarine. A weak submarine would just crunch together. But in this case, this just blows apart. All the little air pockets have to get out at once. Versus in the no-tiller and the native, there's strong PEDs, and then in between those, there are these channels from old root channels that didn't get tilled up, or old earthworm channels that didn't get tilled up. So these allow the air to bubble out slower, and the strength remains. The integrity of the structure is still there. So this is from, this particular slide is from a study that was done at the North Agronomy Farm in Manhattan. And so this is showing that organic matter, so I should say, both soils received the same nitrogen rate for 30 years, which was 135 pounds of nitrogen. And one was tilled and the other wasn't. And another interesting thing is that this was a continuous sorghum experiment that started, probably started when I was in grade school. So, but it was a study that they had a certain purpose for starting this. And I just went in and sampled, a long time later into the length of this project. So organic matter for the conventional tillage was, a little over 2. So let's call that 2.1%. And no-till was more like 3.1%. So that's a percent difference. And that says 7.5 centimeters, which is about 3 inches. So the top 3 inches, there was a large difference of 1% in organic matter. And then below that, there wasn't much of a difference. And that's a pretty common thing we see in the Great Plains, is that our main difference is in the top, but the top is important. The other thing I want to say is that the bulk density of these was very different. So the no-till was less dense than the conventional till in the surface. And then down below, there wasn't much happening statistically, but it is there are some differences here. So the reason I bring up this bulk density is because for any of the carbon credit programs that are being discussed right now for carbon trading or for carbon storage or for carbon sequestration, that's always they're not going to just measure organic matter. They're also going to want to know about the density of soil and the depth. So those 3 things together, organic matter and density and depth, you have to multiply those, and that will give you the stocks. So tons per carbon is how we'll be trading carbon in the future, not percent organic matter change in your soil, but to really know that. So you have to have all those factors to be able to trade carbon and to be able to understand how much you can or can't sequester. So bulk density, when you have, basically density and porosity are the exact opposite of each other. So the more dense you are, the less pores you have, right? So soils that have more organic matter generally have better aggregation, and better aggregation means the soil is less compactable. So if you have a high density in your soil, it could be compacted, but it does depend on texture. So this is just a little table out of a textbook, but it says clay, let's just look at average minimum bulk density that restrict root penetration in soils of various textures. So if you have a sandy soils have higher bulk density because they are overall not as porous as something with more clay in it, which I know sounds really weird. But the thing about sandy soils is that they have less total porosity, but they have a lot of big pores. Big pores, those are the ones that let go of water right after it rains, so they'll drain right out. So big pores are good for water, good for oxygen movement and gas movement, but they're kind of bad for, holding on to water to be available for you later on. So whenever you look at density data, if you do a bulk density sample, which is to excavate a volume, a cylinder or a chunk of soil, if you know its volume, you can determine its dry mass and then calculate its density. You can always look at, okay, is this too dense? Is this just right? You can compare it with textbooks for that. So infiltration, this is that same thing I was showing earlier, the same graph where that had the conventional till had the poorest aggregation. It had the short bar here. Well, now a tall bar is bad. So this says minutes to infiltrate 1 inch of ponded water. The conventional till took 25 minutes to infiltrate an inch of water, versus the rest of these, were ranging from a little over 5 to this Rivech canola said, about 15. And again, this P equals 0.14. As scientists, we like to see that as less than 0.05 before we would start saying, oh, this is statistically significant. So I'm not going to put letters on here to say which ones of these grouped together. But visually, the conventional till was definitely popping out. So it supported what we would think, which is better aggregates equals better infiltration, faster infiltration, in this case being the goal. So I'm going to talk to you about a couple another long-term study. This one took place in Heston, Kansas. So Heston is north of Wichita, but south of Salina. And so central part of the state, and it has 27.5 inches of annual rainfall. This was done on a silty clay soil. Which means it has more than 40% clay, but it also has a lot of smectitic clay, that's the kind that shrinks and swells. So that's the kind of, if you work it in your hand when it's moist, it's like, really hard, and it'll make a long ribbon, but it's really just tough to work. So this was a winter wheat grain sorghum rotation that was done for 15 years. And so after the wheat, they would grow one of these three things, either nothing, so no cover crop, or they'd use sun hemp as a cover crop, which is Crotalaria gensea, or they would use soybean as a cover crop. So it was a late maturity group that didn't put on any seeds. So some alternatives, if you don't want to grow sun hemp, would be cowpea, soybean, or mung bean. So this is showing that after 15 years, the 0 to 3 inch soil organic carbon was very different. And so to convert between soil organic carbon and soil organic matter, you multiply by 1.7. So, and that is because soil organic matter is a little over 50% carbon. So if you ever need to convert between those two, that's your factor, 1.7. And so I'll call your attention to the black bars and these clear bars, these white bars. So the black bars are for the 0 to 7.5 inch depth, which is the top 3 inches. And so if you'll allow me to just fudge a little here, let's just multiply by 2. So 1.4 times 2, so that's about 2.8% organic matter, versus these two, the soybean or the sun hemp, and they both have little A's on top, which means they're statistically the same. This one is about 3.4 and this one's about 3.6. So there was a significant increase in carbon in these soils where we had a cover crop. And the other interesting thing is that we only had a cover crop every other year. So every other year made a tremendous difference. So if you're like, well, I can't figure out where to put cover crops in my system every single year. Well, is there a spot every other year, or is there a spot every third year? I would say that in the most intense cropping systems we have, maybe there's not a window every year, but if you can find a window that makes sense and you can get good biomass production, go for that. And so I'll also point out that again in this study, we didn't see any carbon storage in that next 3 inches, the 3 to 6, just in that top. This is the other figure here talks about the nitrogen application rate and the fact that as we added more nitrogen application, this was to the sorghum crop, as we added more nitrogen, we got more carbon sequestered into that soil. And that makes sense because we grew more crop, we grew more tissue for that crop. And so then it decomposed and formed more soil organic matter. So with more soil organic carbon came more infiltration. So fast is good. In this case, this says cumulative water infiltration in inches and in 3 hours. So we'll just look at the end point here. So 150 millimeters, so 6 inches, right? 6 inches of water where we had the sun hemp infiltrated in 3 hours versus 4 inches or so where we had the soybean versus about 2 inches where we had no cover crop. So that's not bad. No, 2 inches isn't bad, but 4 or 6 is even better. And I think, thinking about intense rainstorms, when you get just a ton of rain all at one time, being able to capture it and then store it is critical, especially on the drier side of our state. But it doesn't matter. Even if you're on the wetter side of our state, you still want to be able to you want to capture as much as you can, because if once runoff starts, then it will erode soils, and that's a problem. So again, I'll point out that this was just with cover crops planted every other year. This is a profound difference in infiltration. And there was also a very big difference in worms. So the units are a little funny here. We converted these and how many you'd have in a yard, a cubic yard of soil. So 3 foot by 3 foot by 3 foot of soil. So where there was no cover crop, less than 100, where there was a soybean cover crop, there was 300, and where there was a sun hemp cover crop, it was pushing 500. So was it the worms causing better infiltration? Was it the organic carbon forming better aggregation, or is it just kind of the combo, all of these things together? And I would point out that we were out there digging these up. This was 2009 in December. We were in grain sorghum stalks. And so the covers had not been living and green and growing for about 13 or 14 months, but the worms were still way higher in number where the cover crop plots had been. So putting them into the system is very beneficial. And here's a Darwin quote for you from 1881. Worms prepare the ground in an excellent manner for the growth of fibrous rooted plants and for seedlings of all kinds. So worms are good for soil and cover crops are good for worms. So it's just a really good system. This is sometimes I'll get asked this question about, do we need to add microbes to soil? And I think about the time I went to a zoo, and this is a Malayan sun bear, and they had smeared honey on the window so that the bear could lick the window and have something to do. It was called enrichment. It was something for the bear to do, but it was also so that people at the zoo could admire its long tongue. They use these and their big claws to rip open logs and get termites or something, insects. But I think about, don't feed the bears, do we need to add microbes to soils, or do we just need to give them the habitat to be successful? I think it's the latter. We give them the habitat to be successful, and I'm not too worried about adding organisms, to every soil. And an economist prepared this graph and concluded that the more productive the soil was, or the better the soil was, it was actually more productive. So this is, there was a nitrogen rate in this study as well. So at the 0 pounds of nitrogen application, having those legumes of the sun hemp or the soybean in there added a tremendous amount of nitrogen and increased the yield a lot. Same for 30 pounds, that wasn't enough. 30 pounds of fertilizer wasn't enough. So having the legumes in there improved it a lot. But the economist said that this boost comes from productivity. And for sun hemp, this is even this up here. So at this 90 pound nitrogen rate, this difference between the top of this green bar and the none is it's a result of the soil being improved. So after 15 years, this was a significant, it was starting to pay back, pay some dividends for yield. So how much carbon can we add to soil? So there's a classic paper by West and Post, and this says 2002, so yeah, a long time ago, but they had analyzed a bunch of different studies and meta-analysis of a bunch of different studies, and they sort of said one of their big conclusions was that going from tillage to no-till could increase carbon by about 20%, or organic matter by 20%. So example, if you had 2%, you might go to 2.4% by switching to no-till. And what if you change the crop rotation and added cover crops? That could increase the organic matter by 10%. So if you started at 2, you could go to 2.2. So what if you do both? That's good, and you'll get more organic matter, but there's not quite a relationship that I know of yet for predicting what that number should be, and it's maybe not quite additive. But there's, again, with the different, all the different carbon trading programs happening right now, they're using some of the same stuff in their equations for calculating how much they predict that you might store. And so I just think that I tell you this so that you have some idea of what's reasonable. But also, results may vary. If this was an infomercial and we were selling a product, there might be but results may vary. There could be examples that are out there, and I know they are out there, where people have increased their organic matter a lot more than this, and that's great. And then there's times where I know of some people that have tried to increase their organic matter and just haven't been able to. There's some variables. So sandy soils, for example, it's really hard to increase soil organic matter in a very sandy soil because it just dries out so fast. Decomposition is fast. So it's harder. It's a little bit easier. And when you have some clay, you can build soil organic matter a little bit faster. But there's a limit there. You don't want too much clay, right? Nobody likes too much clay. So middle of the textural triangle, those are some of the better textures for trying to improve soil organic carbon. So this was a study done at Parsons in far southeast Kansas, so established in 1983 and sampled in 2002. And they were comparing 3 different systems, so no-till, reduced, and conventional. And they found soil organic carbon. Again, if you want to go to soil organic matter, you can multiply that number times all 3 of these by 1.7. But so no-till was about 1.6 versus reduced and conventional till were 1.3 and 1.2%. And if you multiply the carbon times the depth times bulk density, then you get how much storage in tons. So megagrams per hectare is pretty close to tons per acre when you cancel out the metric to English units. But 11.3 tons of carbon versus in no-till versus 8.4 or 8.2 in reduced or conventional till. So that was a noticeable change. So 3 tons or so in that depth range of, I believe that was the 0 to 5 centimeters, so 2-inch depth range over a 30-year period. Bulk densities and aggregate stability, they also measured that. So they saw no-till was significantly better than conventional, and reduced till was in the middle and intermediate to the two, so it overlapped. But statistically, the most significant, the difference was between no-till and conventional. So similar to other studies. So how long does this take, and what else affects soil organic carbon storage? It does take years. I've never seen anything move at all in 3 years or less, and 5, 10, 15 is where you get to a measurable, and especially a really consistent measurable difference out in that 10 or 15 years. And faster changes, like I said, happen in soils with some clay. I don't mean to say, again, don't want too much clay, but some clay. And sandy soils are slow to change. Dry soils are slow to change because more moisture is beneficial to plant growth as long as decomposition is balanced. So in some parts of the world, you could have really rapid decomposition, just breaking down things really fast. Not so much, I'm not too worried about that in Kansas, but it can happen. And so with that, this is where I wanted to end. Oh, I wanted to end here talking about the SLAKES app. This is an interesting app you can get for your phone. And it's an app, and you can take pictures of your own soil falling apart in water. You put soil in a shallow dish of water, and then you hold your phone suspended over. I set it like on a stack of books. I just hold my phone out there, and you watch the soil fall apart in water, and you can give it yourself. You can give it a test yourself. And so I'm trying it out, and it's early, but it's promising. So it's citizen science, at home science, and especially if you have people in need of a science fair project, it's a pretty cool thing. And with that, I'll say that some soils are some properties are slow to change, like soil organic carbon, but ones that are faster or aggregate stability. So if you're looking earlier on, if you're 3, 4, 5 years into a change, that's something that you can see sooner. Immediately stopping or reducing erosion is really important, and fixing that surface. I've said a couple times about how the depth, that we only changed the surface. We didn't change below that. But the surface is everything. If the surface is not right, if the surface is crusted over and water can't get in, it's sort of game over. You have to have the surface right, and that's so important. So with that, I'll take any questions.
Thank you, DeAnn. That is awesome. With the SLAKE app, what does that give you? What are you testing, and what are you
So
seeing?
the way that works is you'll pick out like 3 little pea-sized heads of soil. So little peas of soil, and you set these in like a petri dish, or a white colored dish is fine too. And you put them in, and then you hit start on the app, and it'll measure for 10 minutes and take a bunch of pictures over 10 minutes. And then at the end, it's doing some math, and it gives you like a stability number, and then it'll actually tell you if that's stable or not stable. It was developed by some soil scientists in Australia, and they published a paper, and then one other person has tested it and wrote a paper saying it worked pretty well. So I have a student working on that right now. And so I don't know if it's going to be a cool thing or not yet, but I like that it's free. And so it just costs you your time, and I think it could be fun.
Okay, yeah, it sounds really interesting. So it gives you a measure of how fast your soil is falling apart. Okay.
Yeah, it gives a stability index number of, I think 1 is good and 20, like it says right when the results say like, oh, this is a very stable soil, or this is a very unstable soil. And it has you use three different pea-sized pieces from the same sample, so that you're not just doing one, at least there's 3. And you could do it 10 times with different, PEDs from the same bag of soil to check your results.
Okay, and so you just want a pea-size thing of soil, you put it in there, and then measure this with the app, and
Yep.
it gives you a measure of the aggregation. That is interesting. That is really cool. And you
Yeah.
want more stable aggregates because the house is more well constructed, then, if you will. Okay,
Yep.
cool. Okay, good. You talked a lot about bulk density and aggregation, and those are connected, right?
Mhm. Yes.
Okay.
Porosity, bulk density and porosity. Yep.
Okay, so porosity. And porosity is like a straw. And if you have a big straw from McDonald's versus a little bitty coffee stirrer, that's the relative size of the pore, if you will.
Well, you can have a pore size distribution. So there are people that you can take a column, usually it's a cylinder of soil, and you can wet it all up to saturation to where every pore is filled with water. And then you turn on this suction, and you basically crank it down over time. So you crank it so like at first all the water in the very big pores will run out, and then you turn up the suction a little more, and then so over time, like once you're maybe out a week, you've turned up the vacuum really high, or the suction really high, so it'll pull out. And so from that, then you can make a graph of the pore size distribution. So how many real big pores, how many medium pores, how many tiny pores. And then the other thing interesting about pores is, sometimes they're not very well connected. So if you have a lot of long, nice, continuous pores, that's better than a bunch of just little itty bitty polka dot pores that are not connected to anything else. Long connected pores are far superior especially for fast water movement, for water infiltrating fast. You want those long connected ones.
Okay, and also the pore size and porosity is very important for water holding capacity.
Yes.
Right?
So the most important things for water holding capacity go back to actually texture. So when people talk about available water holding capacity, number 1 is the texture. So how much sand, silt, and clay, and then how much organic matter. That affects it a lot too. But the overwhelming thing that controls it is sand, silt, and clay. Available water holding capacity doesn't have a ton to do with pore size.
Okay. Okay. All right,
But
good.
it does have everything to do with infiltration, capture, and storage, or precipitation, capture, and storage. So like getting the water in, big pores are extremely important, especially big, long connected pores. Storing water, it's important, to have like Goldilocks soil. If you think about the parts of the textural triangle, you don't want too much sand, you don't want too much clay. Silt is wonderful. So silty soils or anything kind of in the middle. So, but any soil can be improved. I will say that any soil can be made better by making better structure so that when rain does come, it gets in.
Okay. And stays in and doesn't just flow all the way through. Yeah.
All the way through, or or with the big killer with aggregates is like if the aggregates fall apart and the soil seals off and crusts, and then everything just goes, and they're like, look, people like, people have a discussion. You're like, oh, it was a nice rain. It was a nice gentle rain. It came slow all day. You're like, yeah, because then it didn't overwhelm the infiltration capacity and seal and run off and cause messes and mudslides, or, just blowing out bridges or whatever. It can be a really rain is wonderful, but we want it to come at our own speed, right?
Yes, yes, yes. Okay. Okay. Yes, there was a comment in chat that aggregation is essential for Southeast Kansas. And yeah, that's what we need to do to be able to capture that moisture and keep it on the field. So
Yeah.
in the field, if you will. Yeah. Okay. Okay, good. Um, what about variability across a field? Does that matter for buildup of organic matter? How large or small your plots are, that sort of thing? How much?
Yeah.
Yeah.
Yeah, I would say, if you think about, so you've got how it was mapped, right? If you think about how the soil is mapped on your field, there's definitely most fields are going to have multiple different soil types. And then soils have changed since they've been mapped. In fact, I got a great question the other day. Somebody said, if you took that line on a soil map, between there's this kind of soil and that kind of soil, how wide is that line? And I did a quick calculation, like that line would probably be, if it was projected down from a projector on the moon, that line itself is 50 feet wide. So soils are variable. They're even more variable than the soil survey tells you they are, because those surveys were done at a certain scale. So precision ag will now allow us to make maps at even finer detail scale, but there is variability, and slopes are one of those natural indicators of variability. So how thick the topsoil is on the side slope versus how thick it is down in the bottom. Each part of that field is going to have a little bit different ability to capture and store water, I think. And so, yeah, with our small plots, we almost always try to put those on really uniform parts of the field. But in reality, there are some places that are going to do a better job than other, in real fields. So I think about controlling runoff is really important. So I've gone to look at problem fields before, and like the biggest problem is the run on coming onto them from another neighboring field. But in the same way, you're almost always upslope of somebody else. So just keeping as much water on your field for yourself so it doesn't become a burden to somebody else is really important. So I guess that brings me to the thing I was going to say, which is this. You've got practices such as no-till and cover crops, but structures are still important too. So terraces, waterways, our ponds are all very important pieces of the pie too, to control, when we have intense rain, that's where they shine and do their work, is when we have 25-year storms or whatever. For them, when we get way too much, we still need those structures too, to do their part of the conservation work.
Okay. Thank you. Interesting. Another question about sampling, and especially when you're sampling for organic matter. Now, we know nitrogen changes variably a lot during the year. If you just put a nitrogen application on, you don't want to measure right then because it's going to be very high. Does organic matter change that much over a year? When would be a good time to get a good measure of organic matter?
So for me, what I always want to do is I always pick the same point in a crop rotation. So let's say that the last time I measured it was in the fall after corn. then I'm going to go back after in fall after corn the next time I measure it. So I try to keep apples and apples. If you measured in the spring versus in the fall, I think, one example I would have is this. So I'll bring out a piece of soil here. When I send in my soils to the soil testing lab, I try to pull out all those roots and all the pieces of residue so that I don't have that's not real. That if you get a sample and you submit it and it's full of residue and it's full of roots, that'll give you a higher number. This stuff isn't really part of the soil organic matter yet. It's just still roots. It hasn't broken down. So that's why I'm really skittish about sampling in, say, spring while the wheat crop is growing and comparing that with fall after the corn crop's been harvested. I guess there's probably corn roots in there too, but I try to keep everything about the same. The size of the microbial pool, I suppose, could be slightly different over the year too. So if you're wanting to look at what kinds of soil organisms are in your crop, it's even more important to do it at the same point. I think of it, it's kind of like, when, like, if you ever have to go in for blood test or something at the doctor's office, like if they're going to check your cholesterol, they kind of have a way they want you to do it. They want you to not eat after, like, within 10 or 12 hours ahead of time. For me, that's how I try to keep it. It's like my fasting soil measurement or something. So, but I do think there is effects seasonally, and I think there are some scientists that are doing that kind of work to, like, make sure that we kind of have the best idea for what is the best recommendation for people.
Okay, thank you. So just really just be consistent. And then it's going to make a big difference. You say most of the changes are in the top 3 inches.
Mhm.
So is that where we need to measure, or should we do 0 to 6?
I know that sometimes for some properties, especially if you measure 0 to 6, you may not be picking it up. So if it's a small change and it's only in the top 3 inches, and you measure 6, you might be diluting that too much. If it's a huge change in organic matter over time, then maybe the 6 inch versus, maybe it'll show up there. But if it's a small change you're looking for, especially, then you get to be careful with the depths. So I think with no-till, I'm pretty sure our K-State soil testing recommendation for fertility says that they want you to split it 0 to 3 and 3 to 6 now and sample that way. So I think that's important for us too. I think that I'm curious to know, like, in the carbon trading world, if that's how they're recommending to be sampled, because that's what all research says, is that depth matters. So I'm sure you've seen that in your own research too, that depth matters for
Very much so.
There.
Right. And I think that's one thing we need to be a little bit careful about in talking about getting carbon stocks into the soil. We're really limited on how deep that's happening. So we've got a huge bulk of soil there, but it's not going down to 18 inches. It's really just staying.
Not very many places. Yeah. And when I'm saying 2 to 3 inches, I am really focusing on Great Plains sort of results. I haven't worked too far outside the Great Plains at all. So, I mean, if there are some other places talking about deep carbon storage, then I'd ask some questions like, well, where is that? And what's the system like? And what are the soils like? What's the climate like? So that I could understand it better.
Yeah. Okay. Interesting. Okay.
On the microphone.
Okay. Good deal. Go ahead.
We've got a farm that on the pastures, the pasture generally is in just excellent shape. But we've got one little pasture that is rectangular, and on the north end, after a burn, there is a very even distribution of rock, like every 3 or 4 feet, maybe a 9-inch rock. I can't wait to get the drone out next year and take a picture of it, because it's going to be something. It goes through 2 draws. There's some water collection from an ancient pond, it seems like. And beyond that, going south, I don't know that we could even burn it because it's so rock oriented, with just the emergence of little, holder honor type grass. Is there anything that can be done to help that beyond just run the cattle on it and graze appropriately. I'm afraid of introducing any seeds from, compost that don't belong there.
And is this in the Flint Hills or where is it?
Yeah, it's outside Council Grove, Morris County.
That's just the hand you have for that soil, probably. Do you think it's very, like, do you think there was past erosion that caused those rocks to be so near the surface, or is this
No, I
just how
think this is really solid rock, and I can add a dose of time to it, but beyond that, I'm just kind of at my wit's end, but I like to experiment. Really managed pretty good, spring water and oak trees to hang out underneath.
Yeah. How deep is the soil? Is it very much on the surface? You don't have much soil?
If there was more grass, there'd be no water there.
I think the biggest thing is if it seems to me you're at the stage of making soil. So like you say, it's just time. Yeah. Okay, good. Good. Well, thank you.
That's a really interesting question. So the Flint Hills have sort of a unique set of challenges. As you said, with the rock that's outcropping there on the edges, that's a natural scenario, a natural system. So our rangelands, there's a reason we aren't farming them. We don't have them in crop production because they are so shallow and so rocky. So as you're doing now with grazing them, that's probably the best thing you can do is to graze them, and a well-managed grazing system, appropriate stocking rates. I don't know that adding more manure or adding any compost there is really going to be very beneficial, just because, as you said, manure and compost, they can have their own sort of issues if you were to introduce weed seeds or something like that. So I think just a properly managed grazing system on top of our very shallow Flint Hill soils that are forming very, very slowly is the best thing you can do for your fields.
Okay.
That work?
You come up with any other things? We start with soil penogenesis. Well, in the beginning.
Yeah, in the beginning.
Good deal. I think this was awesome.